Really a pleasure. My name is York Pei. I'm the director for the Center for Innovative Management of PKD in Toronto. And I am really grateful and honored to have an outstanding panel of PKD experts with us to give us an update on current and kind of practice in ADPKD and also recent advances. And as you can see, this is a hybrid format. Most of the audience are actually in the virtual space, and I hope that they can see us. And if you have any question during any of the session, please type in the online chat box. We will track the questions, and then we will be able to answer them at the end when we have the open forum. So there are 2 days for this conference. The first day today is focusing on clinical management and also recent advances in ADPKD to give clinicians an overview about what is new and what is current to optimize our clinical management of patients. Tomorrow we will have a symposium, and the theme of the symposium is recovering from pandemic. And I'm sure we all have experienced 3 very difficult years. And I hope that we are now closer to the end toward transition to a bit of normality. And the theme of the symposium will be focusing on mental health as well as metabolic health. And we have a panel of outstanding experts talking to us. So I would welcome you to join both days. I also want to thank Otsuka Canada for an unrestricted educational grant, which makes this meeting possible. And please fill in the evaluation form. It's critical for you to give us input, because in the future meeting, what are things you're interested in, and also to give us some feedback. Now I'd like to just take a couple minutes to introduce Jeff Robertson, who is the director of the PKD Foundation of Canada, to give a welcoming remark. Jeff.
Thank you, Dr. Pei. Good morning, everyone in attendance, and of course, joining us virtually today as well. As Dr. Pei mentioned, my name is Jeff Robertson. I'm the Executive Director for the PKD Foundation of Canada. We are Canada's only organization solely dedicated to eradicating polycystic kidney disease. Our mission is to promote programs of research, advocacy, education, support, and awareness in order to discover treatments and an eventual cure for polycystic kidney disease. We are the nation's leader in research and fellowship funding for polycystic kidney disease, something as a grassroots organization we're extremely proud of. What our organization does is we have local chapters across the country that serve as a local support system, as well as people on the ground doing our fundraisers, raising education and advocacy, and supporting patients on that local level. Of course, during the pandemic, things changed, and we hadn't had the opportunity over the last couple of years to get together. So we shifted, as many people did, into the virtual realm. And over the last few years during the pandemic have been holding educational webinars, free virtual webinars that have been extremely well attended, both for Canadians as well as international PKD patients and loved ones as well. I encourage you, those in attendance and those watching virtually as well, to visit npkd.ca. It is the most comprehensive, informative website. It is fully bilingual in English and French. We have a wealth of PKD literature as well as pre-recorded webinars. The last time we were here for the PKD Scientific Conference, that's online. Our educational webinars and the summit is online as well. So there is a wealth of information on a broad range of PKD topics. Speaking of our educational opportunities, our PKD Virtual Summit will be going live for registration starting next week. And this is going to be a 4-evening event on November 16th, 17th, 23rd, and 24th. Each evening has 2 speakers presenting for 1 hour, 45-minute educational presentation, followed by a live Q&A, ranging in a variety of topics related to polycystic kidney disease. Dr. Pei will be one of the speakers as well, joining us, and we're grateful to have his support once again. If you are unfamiliar with the PKD Foundation of Canada, I do encourage you to visit npkd.ca, sign up for our monthly newsletter. It's the best way to stay connected to things happening in the PKD realm, both locally, nationally, as well as internationally. So be sure to do that. And again, if there's anything from a patient standpoint or a clinical standpoint that we can do to support you or your patients and loved ones, please reach out to the PKD Foundation of Canada. We're always here to help navigate individuals on their PKD journey. So again, npkd.ca is our website. My contact information is there. We do have some literature and business cards here as well if you wish to stay connected. Thank you for the opportunity to introduce myself, and I wish you both or all of you an excellent couple of days. Thank you.
Thank you, Jeff. So I'd like to introduce the first speaker, Dr. Terry Watnick, who will be talking about PKD Pathobiology. What do clinicians need to know? You can open the app that is in your link when you register, and that will also enable you to do the quizzes, to answer the quizzes at the end of this presentation. Every presentation will be followed by 2 quizzes, and then we will show you the most commonly chosen answer at the end. Okay, welcome, Terry.
Okay, all right. Okay, thank you, York, for inviting us to Toronto and for allowing us to participate in this conference. So here are my disclosures. Okay, so the mission that York has given me is to update key concepts on ADPKD pathobiology, highlight cyst growth modulation pathways in PKD, and then to discuss how metabolic programming impacts PKD pathogenesis. So this is quite a broad topic, and I have 30 minutes. I will do my best. I'm not gonna talk about every single piece of data. I'm just going to hit some of the highlights. So just because I'm the first speaker, and I know everybody knows this, ADPKD is the most common genetic cause of end-stage kidney disease. It affects about 1 in 500 to 1 in 1,000 individuals, regardless of ethnicity or country of origin. It's characterized by progressive enlargement and development of kidney cysts, and you can see that kidney over there. I don't know, not sure which the pointer is, but you can see it's huge. About 50% of affected individuals will go on to develop end-stage kidney disease, and you can see why. And extrarenal complications include liver cysts and intracranial aneurysms. So ADPKD genetics, about 80% of all cases are caused by mutations in the PKD1 gene on chromosome 16. It's a very large message and protein. The 15% about are caused by mutations in PKD2 on chromosome 4. And then I just want to highlight, and again, I don't have time to go into all the details, but over the past probably like 5 to 10 years, we've appreciated that mutations in a whole list of other genes can also be confused for ADPKD. And I've listed some of those there, and I'm sure that Peter Harris's group at Mayo is probably going to identify more, but that's the list that I have so far. And so PKD1 encodes the Polycystin 1 protein. It's a large membrane protein. You can see the image there. Let's see, is this, not sure where the, do you know where the pointer is, York? Is that? Oh, okay, it may not. Okay, fine. So you can see the picture of the Polycystin 1, a schematic of that protein there. It's a large membrane protein that resembles a receptor. It turns out that proteolytic processing of this protein is important for its function. And those little arrows there show all of the different cleavage sites that have been reported. Perhaps the best characterized one is the one at the GPS site, which cleaves the protein almost in 2, and these 2 halves. So this is an cis autoproteolytic cleavage process. And once the protein is cleaved, the 2 halves remain non-covalently attached. So there have been many proposed functions for this protein, and I've listed them there. I don't have time to go through all of the data, but basically this is thought to behave as an adhesive receptor that's involved in cell-cell or cell matrix interactions. So the PKD2 gene encodes Polycystin 2. It's a member of the transient receptor potential family of cations or trip channels. It has 6 transmembrane domains, and we'll talk about some of this data later, but it's thought to function as a non-selective cation channel. It is calcium permeable, and I've showed calcium there as the ion that it is permeable to, but it turns out it's also permeable to sodium and potassium, and there's some data or some hypotheses that suggest that it participates in flow sensing. So the key thing to know, and I guess this is clinically relevant as well, is that Polycystin 1 and Polycystin 2 interact to form a receptor channel complex, okay? And so that's why the phenotype of mutations in these 2 proteins are identical, because they form a core of a signaling pathway. So whether you mutate one or the other, you're going to get a very similar picture. Okay. So I think the main task that York has given me is to sort of discuss how do we go from that normal kidney, which someone with ADPKD may be born with, to this kind of very large and deformed kidney. And that's where I think a lot of the efforts of the research community have been focused, is on understanding that process, because if we can really understand it, then we could maybe fix it too. So that's what I'll be discussing for the rest of the talk. So the first thing to know is probably when you look at someone with PKD, like if you were to image them when they were an infant, you might not see any cysts, but these cysts develop over time. And so I think, and maybe it's a little bit controversial, that we kind of understand a little bit about that process. So if you were to dissect an early stage PKD kidney, which is what's shown there, that's one tubule, you can see that cysts are not developing in every single location, yet we know that every cell is probably the same or has inherited the same germline mutation. And in between these focal cysts, the tubules, at least to this degree that you're looking at it, look pretty normal. So if you were to be able, and this has been done, to isolate the DNA that lines every single cyst, you can look at that and maybe try to get some clue as to what the first step might be. This was already done, and so I am just going to show that schematically here. So all the way over on the left in A, you can see that all cells in a kidney tubule contain the same germline mutation, right? That's what you inherit from your affected parent. But somewhere along the way, and this is kind of probably a random process, you develop a somatic mutation of the wild type copy of either PKD1 or PKD2 that you inherited from your unaffected parent. And so once you completely lose polycystin in that cell because you had a somatic mutation, you then have clonal expansion of that PKD deficient cell. So it may not be completely missing, but falls below the critical level. You then get tubular dilation and fluid secretion. And so there's some implications of this model. One, every cyst would then be predicted to contain a unique somatic mutation. And then disease severity would then be determined by the somatic mutation rate. And what that means is that it's loss of PKD signaling is what causes disease. So when you have a autosomal dominant disease, you could have, a dominant process, but here it's recessive at the cellular level, and it's loss of function that is causing the disease. So initially, maybe this was controversial because it was felt like, how often does this happen, right? Maybe this is just something that you can detect in a minority of cysts, and the reason I'm talking about this is because it's recent data. So in a study that York and I did many years ago, we were able to detect somatic mutations in about 70% of cysts. So that's not like 100%. So you could say, well, maybe there's other stuff going on. But just last year, another group used whole genome sequencing. So this would be state-of-the-art, very modern sequencing techniques to go back and do that study. And they screened 90 cysts from 24 individual people, and they were able to detect mutations in about 93% of those cysts. And as you would predict, they're all different types of somatic mutations, and that's shown on the right there, just their breakdown of what they found in those 90 cysts. And so I think that you can say in human ADPKD, somatic inactivation of a PKD gene is probably the primary cause of cyst formation and expansion. And that's not to say that there aren't other ways that you could lower polycystin signaling. There probably are, but at least I think it's probably occurring in a very high percentage. And if you could somehow understand why this happens or control it, you probably would be able to affect how severe the disease is. So next, once we've had our mutation and we've lost polycystin signaling, what happens from there? And there are a number of effector pathways that have been implicated in PKD. Okay, I mean, I don't think I have time to go through all of them, and I'm just going to highlight the main ones. And so the ones I'll be touching on are cyclic AMP signaling, which will be covered by Vicente, so I'm probably just going to mention that briefly. I'll be talking some about cilia, about calcium, and then metabolic pathways. So I think it's fair to say that cyclic AMP is a major modulator of ADPKD, and that cartoon that I've shown there is from a review by Vicente showing some of how this is speculated to happen. And so what I would say, and why I think it's a compelling story, is that renal accumulation of cyclic AMP has really been demonstrated in multiple rodent PKD models. And as shown here, cyclic AMP, through a variety of pathways, stimulates the proliferation of PKD cells in culture. And then also cyclic AMP is postulated to stimulate chloride-driven fluid secretion. And I'll talk a little bit more about this. There are several mechanisms by which this might happen, but CFTR, or the cystic fibrosis transmembrane regulator, is postulated to be at least one of the apical channels that might drive chloride secretion and then fluid as well. And then, as I think Vicente is going to talk about, at least in the kidney, vasopressin is going to be the major hormone acting through cyclic AMP in the kidney. So cyclic AMP that's generated through this pathway has the potential to influence all of these different things, fluid secretion and growth, which would promote PKD pathogenesis. And so I'm not going to go through all of that, but and then in my view, there's extensive preclinical data that supports a role for vasopressin and cyclic AMP in PKD pathogenesis. I think Vicente will go through this in more detail, but I've just kind of picked out a few of my favorite studies. On the left there, if you take the PCK rat, which is a model of ADPKD, and you cross it to another rat that's lacking vasopressin, you get worsening of disease, which kind of implicates this as being very important. The next figure over there, if you knock out adenylate cyclase, which is what generates cyclic AMP, in a mouse model of PKD, in the middle you can see the complete knockout. The kidney is big and cystic, but if you knock out adenylate cyclase over there, you improve cystic disease. Then over on the right, these are studies from Vince Gattone and Vicente. If you treat mouse models, and almost any one, with a V2 receptor antagonist, so you're antagonizing the effect of vasopressin, you can improve cystic disease in both orthologous and non-orthologous models. So I think putting this all together, this is an important pathway in PKD pathogenesis. I won't say more about it, and I'll leave the rest to Vicente. And so now I'm going to switch to primary cilia. So primary cilia, it turns out, and we're understanding hopefully more and more about this, are central to the pathobiology of PKD. So for the clinician, you probably haven't thought about this maybe since you were in medical school, but what are primary cilia? So they arise from the surface of most cells, and so I've just picked out from the literature a bunch of different cell types that have primary cilia. They were thought to be vestigial at one point, which means that they had no function. They were just kind of there. But it turns out that these organelles are extremely important to the function of the cell because they organize various signaling pathways, many of which are developmental. So, you know, the classic example would be sonic hedgehog. And the idea is that these little antennae that stick off from the surface of your cell are basically acting as receptors for either chemo signaling or mechanosensors. It can be any of these. So why are these important in PKD? Well, it turns out that the polycystin complex that I showed you in the beginning of the talk, polycystin 1 and polycystin 2, localize to the primary cilium. And you can see there all of the list of various ciliary genes that you can find there. And it turns out that mutations in genes required to build the cilia also result in cystic kidney disease. And this is just one example. KIF3A is a motor protein that's involved in making sure that the cilia is built properly. If you mutate that gene in the kidney in mice, you get a cystic kidney, and that's shown there. So all of this together implicates cilia as being very important in PKD. And so I'm bringing this up. This is more recent data. So, you know, you'd think that if you knocked out cilia and a PKD gene, you'd get worse PKD, or at least that's what I would think. But that turns out that that's not the case. So over on the right, you can see a PKD1 kidney. So if you delete PKD1 in the kidney of a mouse, you get a really big cystic kidney. If you delete like KIF3A or cilia, you also get a cystic kidney. It's just not as big. It's not quite the same. However, if you mutate both in the same mice, and that's shown in the middle there, no cilia and no PKD1, in fact, you get some improvement in the PKD phenotype, and that's shown by that middle kidney there. So what is the model? So the idea is that if you have the polycystin complex in cilia, it's repressing a pathway in the cilia. If that's all working properly, you get no cyst, right? Whatever is causing cystic disease, you have polycystin there in the cilia, it represses it. If you lose polycystin in the cilia, all of a sudden, whatever it's restraining is unrestrained and you get a cyst, okay? If you lose the cilia as well, whatever that pathway isn't active anyway, so you get improvement. That's the idea. And if you could understand what that pathway was in the cilium that's being restrained, that would potentially be a target for therapy. So there's a lot of work going into trying to figure that out. To my knowledge, I don't think we know what that is yet. So, okay, cilia are important, but what is the polycystin complex doing in cilia? So one hypothesis is that luminal flow. So in the kidney, you have these like little cilia sticking off of the epithelial cells, and they're in the urinary space, and urine is flowing by there. And so one idea is that polycystin 1 at the tip of the cilium or in the cilium is acting as a mechanosensor. So it's like sensing the fluid go by. And this fluid flow then bends the cilium and results in calcium influx through the polycystin 2 channel. And so the idea is that this signaling that's going on when it's working right is suppressing growth, inducing proper differentiation of the kidney, and then maintenance of tubes. And that below is just a schematic from some data showing that you can influence flow and you get calcium influx. So it turns out, I think it's a lot more complicated than that. And so this is the updated data. So I think that it is true that polycystin 2 is a channel in the cilium. And so this is work from David Clapham's lab, and I just really like this because I think it's so cool. That's him patching, putting a pipette tip on this very tiny little cilium and measuring calcium current. So the technical advance to get this it's hard. So what he found is, and I'm not a patch clamper, so I don't expect you, this is like hot, gonna talk about this in a way that I think you can that everyone can understand. But if you take IMCD cells, which are kidney cells, and you treat them with shRNA, which knocks down different proteins, you can see that when you patch clamp cilia, only shRNAs against polycystin 2 are able to decrease the current that can be measured from cilium. Okay, so interestingly, you'd think that if you knock down PKD1, you should also get that, and they didn't find that. So here, PKD1, there's no change in current, and in these two there are. Okay, so that's from cells where they used shRNA, but if they isolated renal epithelial cells directly from mice where they had knocked down PKD2 and PKD1, they got the exact same result. So you can see here, PKD2, in those cells, there's decreased current in cilia, but in PKD1, there isn't, okay? So, you know, it would suggest that at least polycystin 2 is a calcium channel in the cilia, but it turns out it's even more complicated than that, because although polycystin 2 is permeable to calcium, it is also permeable to other cations. And so when they looked at this using their system, the channel permeability wasn't like calcium was the most, it was actually the least. So the channel is most permeable to potassium, then sodium, and last calcium. And so it turned out that in these studies, inward calcium, like calcium through the polycystin 2 channel, only happened when the cilia membrane was hyperpolarized. So in other words, when it was very, very negative. So if you have a very negative environment inside the cilia, that's going to be a draw for calcium just based on the negative charge. And so what they found is that under physiologic conditions, there's going to really be very little movement of calcium into the cilium because it's not that negatively polarized. You have to actually make it more than the negative 18 millivolts that exists. However, if the ciliary calcium concentration rises, then that does increase the open probability of polycystin 2, presumably to calcium. So if you can somehow increase the calcium concentration in the cilium, then that would promote calcium entry. So it's not that clear cut, and I think we can say under physiologic conditions, I think it's not still 100% clear how this works. And then just to get back to that original question as to whether flow is what causes this increase in ciliary calcium, again, from the same group, they managed to isolate tubules and like patch clamp them or do calcium imaging. And when they induced flow, like in the lab, you get like this little tubule, you push flow through it, and that's what they're showing there. That actually didn't happen. So mechanical forces didn't evoke calcium signaling in the cilium of either IMCD cells, which they isolated, or intact kidney tubules. So I don't think that says that calcium's not important. Flow may not be just the major regulator of this, but instead, one hypothesis is that if you increase cytoplasmic calcium, this can then flow into the cilia, and then that increased calcium concentration would open the channel. So that's where I think we stand there. And then I just feel like I have to highlight this advance, which is that the complexity of this system has actually, in my view, been revealed by cryogenic electron microscopy. So that's like where you take the proteins, you freeze them so that you can actually look at them and the actual structure under the electron microscope. Okay, I'm sure it's much more complicated than that, and so people have done this, and what they found is that you can assemble polycystin 2 into its own channel just with polycystin 2, and then there's another complex, which is the one I discussed, which is polycystin 1 and polycystin 2 together. And so on the left there, B and C, that is the cryo-EM, or what they came from doing cryo-EM, the model that they were able to draw. And what they found is that the polycystin 2 homomer, meaning all of the polycystin 2s going together, they form a tetramer. So you get four polycystin 2s making the channel. There's a pore between the 5th and the 6th transmembrane domains. And then just to highlight the one very unique finding was that between the 1st and 2nd transmembrane domains, Polycystin 2 has this very big loop, and they call this the top domain. And it turns out that this is like quite an important domain in the protein. They have speculated it kind of sits like a lid over the pore. And so it's been speculated that this is kind of gating the channel. And when you look at mutations or missense changes, amino acid changes in Polycystin 2 that cause disease, a lot of them are located in this top domain, suggesting that it is important. So on the right is the PKD1, PKD2 channel. And so that this is also the cryo-EM structure. And what they found is that in this heteromer, they assemble in a 1:3 ratio. So you get 1 PKD1 and 3 Polycystin 2s, and they form a structure. And I kind of skipped over it, but the last part of Polycystin 1, the C-terminal 6 transmembrane domains, actually have homology to Polycystin 2. That little picture there shows it. But so it does have some, like structurally, you could imagine that it may be a channel. So, but this kind of forms an anomalous channel because when Polycystin 1 fits in there, it kind of fits in at a funny angle, and the top domain doesn't quite fit. And most importantly, it contributes some positive charges to what would be predicted to be the pore. So positive charges there are going to prevent positive ions from flowing through. So it isn't clear whether this kind of heteromer is active or not, or whether what could it maybe abrogate activity. So I think, although this is, I think the latest, I think it also opens up more questions that we need more work to address. So now I'm going to get to metabolic reprogramming. It's a huge topic, but I'm going to highlight a few areas that I think are important, maybe clinically. So this kind of started this idea that ADPKD was associated with metabolic disturbance, started with some observations from Alessandro Bolletta's lab in Italy. And so this is, I like it because it's just very visual, but if you grow mouse embryonic fibroblasts in a dish, PKD1 mutant cells, you can see there on the left that the knockout cells, just the media, is a lot more yellow, right? And so these media that you grow cells in have pH sensors, so it's doing that because the pH is lower. And so when she analyzed the media, what she saw is that the glucose level in the media was a lot lower in the PKD1 null cells, and that's because they're using it up, so there's less in the media. And so you can see that over there on the left, but what was higher was the lactate level. So what was going on is that these cells are using a lot of glucose and basically converting it into lactate, which is what's making the media more acidic, and it looks yellow. Okay, and she looked, and I'm just pulling out one piece of data there at glycolytic enzymes and saw that they were upregulated in these cells. So what is going on? And so this is, you're going to need to take out your Leninger from biochemistry, but this is what she hypothesized, is that these cells were like very dependent on glycolysis. And this is called the Warburg effect. And it's something we see, or that has been reported in malignancy, to be associated with malignancy, which is a proliferative process. You have too much growth. And, you know, you could say that PKD is the same in that regard. Obviously, it's not a malignancy because the growth is not that fast and you don't have metastases. But what she saw was that PKD1 deficient cells were dependent on aerobic glycolysis for energy. And so just to remind you what that is, so this is a little cartoon taken from a review paper, but on the left you have, quote, normal cells. And normally what we were all taught is that cells take glucose and they metabolize it to pyruvate. And when oxygen is plentiful, then the pyruvate is oxidized to CO2 via the TCA cycle, and then you have ATP being formed by oxidative phosphorylation in mitochondria, right? That's what we were all taught. Then when you don't have oxygen, like maybe if you're exercising a lot, the pyruvate, because you don't have oxygen around to go through all that, you're then metabolizing it to lactate, right? However, in cancer cells and in ADPKD cells, what they do, and even if oxygen is around, they convert all of that glucose to lactate, regardless of oxygen availability. And if you do that a lot, you actually can generate a lot of ATP. And so from a cellular level, like, why would you want to do that? And the idea is that you're diverting glucose metabolites from energy production to anabolic processes that are also required for growth, right? If you're going to grow a lot, you need more than ATP. You need ATP, but you need all those building blocks. And so that's what's going on here. Okay, and so what she was able to show is that if you could inhibit glycolysis, this whole process, that you could improve PKD in mice. And so what she did, and this is a series of papers, is she used 2-deoxyglucose, which is a glucose analog that can't be metabolized. Okay, and so what you're seeing over there on the left is that she treated mice with this 2-DG, and you can see that if you treat with 2-DG, you get some improvement in the size of the kidney. So all the way over on the left, the vehicle, sodium chloride, the kidney's very big and cystic. And then in the middle where you treat with 2-DG, I mean, it's not quite control, but it's better. And then in the middle panel, we're looking at the total kidney volume. And by 2 months, if you go all the way over to the right, you can see that the 2-DG treated mice, the kidneys are not as big. And then maybe the best for clinically looking at it, doing MRI in mice. The bottom is the treated mice. You can see that the kidneys are not as cystic over time. So if you think that this process is important in pathogenesis, there's also some evidence that if you can reverse it or slow it down, that it also improves PKD. And there is going to be a clinical trial, I think, of 2-DG that's going to be starting. So this whole process is actually also related to mTOR signaling. And so for a long time, people kind of just wondered whether mTOR signaling would be important for PKD. And I don't know, there's some sort of data that suggested this might be the case. Maybe the major one is that if you ever have seen patients with tuberous sclerosis, there's a subgroup of them that also develop kidney cysts. So there was always an idea that there might be some relationship. And also it turns out that the TSC2 gene and PKD1 are right next door to each other. So people who have deletions of both of these genes really have a very severe PKD. So this is kind of observational stuff, but Thomas Wimes, who's here, went on to look at this and more. Am I getting close? Okay. So mTOR senses cellular energy and is pro-proliferative. I just want to say a little bit about mTOR. It's a kinase that forms the core of 2 complexes, but we're just going to focus on the one on the left. And it's an energy sensor, okay? So there are many inputs, positive and negative, into this whole system. So it's confusing, but in PKD, the loss of polycystins, the idea is results in an increase in MAP kinase and ERK. And if you look over there on the right, ERK is one of the things. So TSC1 and TSC2 are negative regulators of mTOR. So if you get rid of those, you're going to increase mTOR. So ERK decreases the activity of TSC1 and TSC2, and that results in an increase in mTOR activity. And so that's the idea. And so basically, down there is what I want you to remember, is that the availability of energy sources, like if you have a lot of energy and stuff, you're going to activate mTOR because you have a lot of stuff to fuel growth. However, depleted energy sources turns it off. And so this is just showing you some data that mTOR is upregulated in PKD. And this is looking at human kidneys, PKD1 mutant mice, and just showing that if you stain those for mTOR activity, you can see that it's increased. And then I just want to quickly say that this mTOR signaling is also very linked to AMPK, okay, which is what's activated by metformin. So this on the right is from Alessandra Balletta's paper, just showing how she speculates this all work. So I just told you that ERK1/2 is increased in PKD. That will increase mTOR, increase glycolysis. It increases ATP, which turns off AMPK. Okay, so it's complicated, but bottom line, too much mTOR, too little AMPK, and this is what might increase growth and increase glycolysis. So if you treat mice with metformin, which increases AMPK, a mouse model, it does seem to have some improvement in cyst formation. And I'm just going to say briefly why, again, getting back to all of these pathways, why activating AMPK might be beneficial in PKD. So the idea is there's a negative feedback loop. If you increase AMPK, you're inhibiting mTOR signaling, which we said was increased. It also phosphorylates CFTR, which would inhibit its activity. And we said that CFTR might be one of the chloride channels that promotes fluid moving into cysts. And then it could also activate phosphodiesterase, which would decrease cyclic AMP. So for all of these reasons, if you activate AMPK, it might be beneficial in PKD. And just very quickly, I'll just show you, this is a mini pig model. So it's a pig with PKD. And if you treat the pig with either 2DG or metformin, and that's shown on the left there, you can see that you have like a big improvement in cystic disease. So last, I'm going to get to lipid metabolism as being a modifier of PKD progression. And so again, over there is a summary in case, I had to look back at this. Basically, to remind you, there are 3 pathways for ATP generation, right? So you can take pyruvate, you can take fatty acids. It can go into you get acetyl-CoA, it goes into the TCA cycle, and then you get oxidative phosphorylation, right? So fatty acids are one of the substrates for this. And so this is data from Greg's lab showing that if you do seahorse, which you give it a fatty acid as a metabolite, that you have decreased oxygen consumption. So you're not using fatty acids well in these cells. And so he made all of these different cell lines for mice, some lacking PKD, some having it. And the solid lines are the wild type, and the dotted lines are the mutants. And in every case for each pair, the wild type cells use fatty acids much better than the mutant cells. And then again, just showing here other data. This is from Vishal Patel's lab. It turns out that PPAR alpha, which is what's regulated by like fenofibrate, is it's a transcription factor. And the targets of this transcription factor are all genes that are involved in fatty acid oxidation. Okay, so this is showing in mice that the levels of PPAR alpha are decreased, as are all of the downstream genes. So this would be one explanation as to why you don't use fatty acids as well, because this very key transcription factor is downregulated. And okay. So just if you can increase PPAR alpha using fenofibrate, which is a drug that we use to treat hypertriglyceridemia, you can see that you can increase on the left there the level of PPAR alpha, and you then likewise increase the transcription of all of the downstream targets in the lower panel, and then you have some decrease in cyst formation. Okay, so and this whole pathway, all of what I've been talking about, would lend itself to dietary modification, right? Because you can affect a lot of metabolism like by fasting. And this is something that Thomas is going to talk about more, but this is just sets the table for it. Okay. And then I got 2 more slides. So knockout cells, so all of what I've been telling you about metabolism, all involves the mitochondria, because all of these processes like the TCA cycle, oxidative phosphorylation, all of that is taking place in mitochondria. If you look, and this is plenty, other people have done this, I've just pulled one piece of data out, but if you look at mouse renal epithelial cells, control and mutant, so these are mitochondria. You can see in the wild type cells, they look kind of nice, and you see all the cristae going in there like we all learned in histology. But if you look at mutant cells, they're just, you can just see it. I don't think you have to quantitate much, but it has been, but they just don't look right. They're little, the just doesn't look right. I think the key question here, because these are already cystic animals, is whether this would occur prior to cyst forming, and I don't think we know that yet, but okay. So to end, in summary, PKD results when polycystin function or signaling falls below a critical threshold. I think that it's fair to say that the PKD proteins are part of a complicated nexus of signaling pathways, and many of these pathways, and it gets complicated, are interrelated and kind of involve positive and negative feedback loops. And cyst modulation factors that I think are important include cyclic AMP, calcium signaling, and cilia function. Altered metabolism, as it turns out, appears to be a major driver of PKD, and some of the metabolic processes that have been studied and identified are enhanced glycolytic flux, increased mTOR and decreased AMPK, decreased fatty acid oxidation and utilization. I didn't even get to it, but there's others that are being worked on, including altered glutamine metabolism. And so I think why this is so important is because targeting metabolic pathways could potentially modulate disease progression. And that's what we're going to be talking about, I guess, in the remainder of the talks. So I'm going to stop there. Yeah. I don't know if do we do questions now or no questions? Oh, the quizzes. Okay.
So there are two questions from Terry's talk.
The third choice is Polycystin 2 is a non-selective cation channel. There's a membrane of the trip.
Okay, so the second quiz question is, which of the following is true about metabolic dysregulation in ADPKD? A, altered glucose utilization and fatty acid oxidation has been implicated in ADPKD. B, treatment of ADPKD mice with 2-DG, a glucose analog, does not improve cystic disease. C, increased AMPK activity could slow PKD progression by inhibiting mTOR signaling and CFTR, thereby decreasing fluid secretion. D is A and B, E is A and C. Okay, can we show the results, please? Okay, so most of you have it right. The answer is E. Okay, so now I'd like to introduce the next speaker, Dr. Ron Perron from Tufts University in Boston, and he's going to talk about management of